we need inventory of regional biodiversity. We have, of course, barriers and challenges to face, the knowledge gaps. There is not that much known about these species, not about the ecosystem they are in. We want to support this green market development. It's hard, but we want to do that and try to provide a label of green market. And of course, we have financial constraints to continue a project, but we hope to navigate through all of that. And okay, with this, I would like to thank you and all the collaborators. This project is funded by the Ministry of Science in a program for women in science and for bioeconomy and territories. And I have a great collaborator team of universities in Medellin and a great support in the field from the NGO and the reserve. Thank you for your attention. And I don't know if there's time for questions. Yeah, so right now, what I have done is, so we have the leg of the bee, and we already did the morphological characterization. And we're going to sequence that through Sanger, like to have a sequence of the cytochrome. And then what we want to do is to do a high throughput of the honey, which I'm now doing DNA extractions. And I can tell you, indeed, it's quite complex because all the inhibitors and stuff. But then run the cytochrome, the tRNL, 16S, and ITS from the honey. And because we also have from those same samples morphological data from the pollen, we kind of want to do some, I know it's difficult, but some sort of match, at least of the big taxa group, of how does eDNA match to, for example, morphological data. And the same with the fungi and the bacterial. So try to approximate it from two perspectives, the morphological and the eDNA. We already have challenges because indeed their references for plant and for pollen are difficult in some areas, especially in these like high diverse, remote, like where you can only get to one family, but that family can be like who knows what. So indeed, and I already anticipate that with the eDNA, we're going to have something similar. I mean, we probably, with the reference that we have that are very little and very poor, we're going to get to maybe some family, if we are lucky, once in a while to genus, but thank you.
Hey, good afternoon, everyone. So I'm Peter Moorn. I'm from Naturalis Biodiversity Center in the Netherlands. Today, I want to give you a quick overview of the Antenna project. So Antenna is developing an integrative monitoring framework, combining novel technologies and traditional methods to address critical gaps in European pollinator monitoring. Next slide. Okay, perfect. So pollinators are declining across Europe and globally. So in Europe, up to 35% of hoverflies are threatened with extinction. But the true pollinator crisis is unclear because there's massive amounts of missing data. So for the majority of species, we lack population level data. So we cannot actually assess the species status. For example, in Europe, which is probably one of the best studied regions, up to 79% of wild bees lack trend data. And this is associated with monitoring bottlenecks related to traditional methods, which are quite slow, costly, and highly dependent on expert taxonomic skills, which are quite limited. And this leads to massive taxonomic, geographic, and temporal gaps in our data. And so this figure here shows sampling incompleteness across the EU for wild bees. What we can see is the regions that are darker have more missing data. These regions, coincidentally, are also the regions with the highest biodiversity of insect pollinators. And so this highlights a critical knowledge gap that conservation decisions are being made without the adequate data. And so in response to this, the EU set up the European Pollinator Monitoring Scheme, also known as EU POMS. The aim of this is to standardize pollinator monitoring across the EU. And through a number of pilot projects, it's been recognized that in order to scale up monitoring across the EU, novel technologies are essential. And this is where the Antenna project comes in. So Antenna is a 3-year EU project across 6 countries. These are listed here. And the goal of the project is to develop and test technologies that can overcome these monitoring bottlenecks. So we can have a comprehensive monitoring framework across the EU. And so we're advancing 4 key technologies. AI camera traps in the field, so shown here on the top left. These are a Diopsis and a Minimon camera, so we're testing these in the field. Then drone mapping for habitat quality, and then automated sample sorting and DNA-based methods in the lab. And so we're testing these across different ecosystems and quantifying how they compare to and complement traditional methods. So the idea is not to replace traditional methods, such as transect walks, shown here by the net, and pan traps, but to quantify the added values of these novel technologies and how they can complement them. Bear with me. Yeah. 1, 2 more. Yeah, I think that would be easier. Yeah. Okay. So here it's perfect. Okay, and so the Antenna project is structured into 5 integrated work packages, and I'll just give a quick overview of these. So work package 1 and 2 is about advancing and testing these new monitoring technologies. So at Naturalis, we're currently developing DNA metabarcoding workflows, and metabarcoding is really good for getting rapid species identification, especially if you have good DNA reference databases. However, a critical challenge is that metabarcoding cannot give you reliable species level abundance data, which is essential for the population trends that we need. And so one solution that we're looking at is integrating AI imaging, automated sample sorting into barcoding workflows. So just to give you an idea of what this might look like. Next slide, perfect. If you start with a bulk sample collected in the field using like a pan trap, and then you can use automated sample sorting. So initially, we're currently testing this using manual sorting. So we're sorting bulk samples into the major pollinator groups. So for example, bees, butterflies, hoverflies. And then we're going to image and barcode every specimen. And this will be in the initial training phase. And then using imaging, we can train AI models to identify specimens. And once specimens can be confidently identified, we no longer need to barcode them. So we can use imaging as a filtering approach. And this will give us specimen counts, species identifications, and also body size estimates, which are really useful. And then for specimens that cannot be confidently identified based on morphology, we can still use barcoding or metabarcoding workflows for them. And so this is very much under development right now. This has been led by the group in Denmark, who are focused on the AI imaging and the automated sample sorting. And so work package 3 is then integrating all this data from these different monitoring approaches to develop forecasting models. So we can actually have an early warning system where we can detect changes in pollinator populations. This part of the project is being led by the group in Spain. Back 1, yeah. And then finally, work package 4 and 5 is putting together everything we've developed and scaling it up. So going from local demonstrations to large-scale transitional transnational pipelines. And we'll be able to offer practical guidance on which methods work best in different contexts. So this will be really useful for different countries that face different challenges, as I showed across the EU. There's a lot of different challenges comparing Greece or Spain to the Netherlands in terms of the taxonomic diversity and reference databases. So we'll be able to offer practical guidance on which methods work best in different contexts and how complementary methods are, while ensuring that data is comparable and standardized. And with that, I just want to thank all the colleagues involved. So this is the project leads from the 6 different countries. And then I also want to thank you all for listening. And I'm happy to take any questions.
So really nice project. I was wondering if you did not consider using genomics since you're talking about population trends.
Yep. So absolutely, genomics is going to be a key part of scaling up pollinator monitoring. That is an area that we're looking into at Naturalis, and it is part of the EU pollinator monitoring program, has identified population genomics as essential. So that is something we do want to look at. I think it'll be really challenging because a lot of population genomics and conservation genomics has been developed for vertebrates, and some of these pollinators will have huge effective population sizes, and they differ quite a bit in their reproductive strategies. So coming up with effective measures or EBVs will be an interesting challenge, but that's something I definitely, I'm interested in and be happy to discuss more.
Okay, so the next speaker is Sophie Pamerlon. She's going to talk about eDNA, data structuration, and good practices for public policies.
So good afternoon, everybody. I'm Sophie Pamerlon, and I'm sorry my voice is not very loud, so if you don't hear me, you can make signs for me too, and I'll try to speak louder. I'm from JB France, and JB France is hosted at the National Museum for Natural History in Paris. And I've been working for roughly a year and a half with all the colleagues that you can see here from the museum too, around eDNA data structuration and good practices for public policies, because there's been a huge demand in France from researchers or protected area managers that already worked with eDNA techniques to share this type of data, and our information systems and the national databases were not able until very recently to handle this type of data. With so my plan will be the following. I will share some context and the goals of the working group that we put in place in order to work on these topics. Then I will show you some deliverables from this working group. So the short practical guide for eDNA-derived data publishers, and an eDNA template that we've also created in order to integrate this type of data into our national information systems. I will also mention the O2 and ASV table publishing through the JBIF Metabarcoding Data Toolkit. That is a new tool that was recently developed by the JBIF community. And finally, I will talk about some conclusions and perspectives and feedback from this year and a half of working on eDNA data. Oh, sorry. So the first thing that we didn't want to do when creating this working group at the museum was to reinvent the wheel. That's why we've been heavily relying on existing guides and documentation tools that were already present and shared within the international community, starting with the GBIF OBIS Best Practices Guide to publish DNA-derived data, the GBIF MDT test program that we volunteered at GBIF France to be part of. So we, since a few months now, we have our own MDT instance to share eDNA data in the form of O2 or ASV tables. And it's been already used, and we showed you that data. And we've also been following, of course, the TADWIC interest group on environmental samples and eDNA that was, I think, presented yesterday by Miwa Takahashi, especially the recommendation of the FERRI initiative in order to share the best data and metadata possible in the context of eDNA data. So for a bit of national context now, our government, I mean, the one we had in 2024, because it's changed like roughly 12 times in the meantime, if we've been following French project. But anyway, our government issued a national biodiversity strategy document that includes a large-scale biodiversity survey through eDNA and DNA-derived data. So this program, this survey, is included in the governance of our Natural Heritage Inventory Information System. So that's a big name for the acronym SINP. Yes, it seeks to be complementary with classical inventory techniques with the help of eDNA and DNA surveys. So the objectives of this national biodiversity strategy and the eDNA DNA survey are to reinforce knowledge acquisition programs, to define priorities and opportunities together with experts and coordinators and data managers, and to highlight actions that were already undertaken by integrating the many existing initiatives, including research projects, because in France, research and public policies sometimes are a bit separated. So it's, we're trying to create links between the two communities. And we wanted to, I mean, the government wanted to give priority to overseas territories, especially French Guiana as a pilot territory. So there's been a lot of inventories and surveys and eDNA data collecting in French Guiana, and there will be more in the future. Thank you. We also have links with the National Sequence Reference Database, GenRef, that was also presented yesterday by my colleague Liliana Ballesteros from the MNHN, the museum in Paris. So I won't say a lot on the subject. I just invite you to look at her presentation, and I think a video will be available soon. This is, sorry. Yeah, this is her presentation. So if you want to know more about the National Reference Library for sequences, you can have a look at this presentation. Okay, so in this slide, I will very quickly explain the eDNA metabarcoding data acquisition workflow. I think you already all have seen similar representations of all the steps that are necessary to acquire eDNA-derived data. They can be roughly separated into 6 main steps. The sampling event with a collection of sample materials. DNA extraction that gives you DNA samples. DNA amplification, DNA sequencing, where you get the raw data. Bioinformatics and analysis, where you get analyzed data such as FASTA, and then the results of the taxonomic assignation, where you get what we call the validated data with the taxon names. Just to wait, it doesn't work. Sorry, so let me just try to go back. This is my, okay, so I'm not finished yet. Okay, this one. So friends being friends, we have lots of national databases and information systems and repositories and catalogs and data portals. So we know that the biodiversity data landscape is a bit complex to understand for data publishers. So we know the starting point of our working group was that there were lots of questions asked by these data publishers, even outside of the eDNA community. And the main question were where to host and share the data. And in the context of eDNA, which kind of data were we talking about? Because as you saw, there were data coming from every step of the process. And where should we share them and how? In which information system? So you can see here the schematic representation of all our information systems. So if you have data coming from public policies, we were first wondering how to get them into the national information systems and then to the international scale. So our main goals were to simplify the integration of eDNA data and metadata into a fair framework, adapt the standards in the context of French biodiversity information systems, because in France we use another standard than Darwin Core, and anticipate and size the data workflow for each information system. So for that, we published a short guide for publishers that lists all kinds of information that we deemed mandatory, highly recommended, or optional, in order to collect data and share data that were the most as exhaustive as possible. So the main public for this guide is the French data publishers that use eDNA techniques to acquire data. Trying to be a bit quicker because I don't have much time left. So in this guide, we have 4 requirement levels for the metadata and data. So as I was saying, some of them are mandatory, some of them we deemed essential in a scientific way, but not in a technical way to integrate into database. Strongly recommended information and optional information for each of the 5 parts of the process. I mean 6, but we merged some of them. If we can get something. This is just an example of it. Okay, so the main goal was for data publishers to submit the data in one single place. So depending on their scope, they could submit either to the national information system or to GBIF or to a thematic or institutional repository, but everything is interoperable. So we really wanted to avoid duplicates, and so people who get eDNA derived data can share them in one single place, and then it gets shared with the other information systems. So I will maybe skip this slide because it's a bit long, but we've been creating also an eDNA template for integrating the eDNA derived data directly into the national database. And it's complementary with the MDT meta-backcoding data toolkit developed by JBIF and that we installed at JBIF France. So when publishers have eDNA derived data, if they have their data as O2 or ASV tables, they can use the MDT. And if they don't, they can use the eDNA template for integrating the French database. And when they use the MDT, the data is published to JBIF and then harvested by our national database and integrated into the database. And, sorry. Okay, so this was a sign for me to finish. I just wanted to show you an example of a data set that was published by a French publisher through the MDT, and it worked really well. So this tool has been really it's really good, I think, for the French community, especially the researchers. And thank you. Skip to the set. Okay. And Rachel Haderle, who is the main author of the data set, is also part of the FAIRY initiative from the Tadwick interest group. And with this, I will say gracias. And just say that, sorry, I will just finish by saying that we are continuing this work and following all the international best practices and evolution of standards, such as the Darwin Core Data Package, in order to better improve our national workflows. Thank you. Yeah, okay.
Maybe I can start by introducing myself. I'm Angela Fuentes Pardo. Okay. There you are. So I'm originally from Colombia, working in Sweden. So I'm very happy to be here. It's actually a dream come true, to be honest. La presentación va a estar disponible en español. Al final van a tener ustedes un QR code en el cual pueden tener acceso a todas las slides, la presentación al final. Y va a estar también el QR code para todo el GitHub y demás. Now let's back to English. So I'm delighted to introduce you to the Swedish Reference Genome Portal, which essentially is a hub for non-human eukaryotic genomic data visualization, discovery, and reuse. So this is a collaboration between Science for Life Laboratory, also known as SciLife Lab, which is a national infrastructure in Sweden that provides e-infrastructure facilities, compute, different data producing facilities, that support life science researchers in Sweden. Also a collaboration with the Swedish Natural History Museum, as well as the National Bioinformatics Infrastructure Sweden, also known as NBIS. So why should we care about genome assemblies and genome annotations? Probably this is the best crowd, and probably I don't need to go into much detail on this, but essentially, these are the backbones to rebuild genetic diversity and inbreeding. For example, also these data help us enable species and hybrid identification. It can guide conservation and breeding programs, and also it can help us monitor genetic health and adaptation. So ultimately, by studying genome assemblies and genome annotations, is not only a need for the academic community, also it serves a wider, oops, there was an animation there. Maybe this is the old presentation. But okay, let's go with this. I know what I changed, so it's fine. So essentially, it serves a wider community by informing conservation and biodiversity protection efforts. So, however, access to these kind of data is surprisingly limited. And the reason is because often data sets can be scattered across repositories, can be hidden in supplementary materials, or even never get published. So in some cases, when it's actually available, can we change to, is it possible to change to the other presentation? Because, I mean, the QR codes and everything are there. I'm sorry. Sorry about that. I just added the QR codes and everything, and that's in the new version. Okay, sure. Okay, so let's see. Okay, great. Yes, so let's move on. Yeah, Yes, so if you click, then, okay, so I just said that, so next one. Okay, so as I said, however, access to non-human genomic data is still surprisingly limited because data sets are scattered or hidden in supplementary materials or never get published. Next. So even when available, they can be large, complex, and could also be technically demanding. So as you could imagine, this creates some barriers for people lacking bioinformatics expertise. Click. As a result, essentially, we are in a situation in which valuable publicly funded data remains underused. And this is particularly the problem that we want to tackle. Next. So here I introduce you to the Swedish Reference Genome Portal, which offers a solution to what I just said, problems with access to genomic data. But in this case, it's targeting the Swedish community. But I just want to show that this is an example that worked for us, but also can be replicable to anyone, any other institution or research group. So ultimately, this is a web platform that provides easy discovery, visualization, and sharing of non-human and curated genomic data. If you have your cell phone here, I invite you to take a look at the website. Next. And for every single species entry, it has 4 different web pages. The first one is the description tab that essentially summarizes some contextual information of the species. You have a occurrence information data that is retrieved directly from GBIF. You have information about the taxonomy and a species photo. In the genome assembly tab, you have more information about the genome assembly itself in the technical terms. So some statistics about the completeness of the genome, but also you can have the scientific article that might be describing how this genome was produced. In the download tab, you have a collection of all the links. So essentially, you can go to that tab and download all the source files. Because ultimately, here, we are just aggregating data that is already available elsewhere. However, we accommodate situations in which researchers, for example, they're working on a manuscript and they want to have their species displayed here in the genome portal, so we can accommodate a situation in which there is a privatization when we upload the files and build the web page for them, but it only will become live when the data gets published in a domain-specific repository such as ENA or NCBI. And the core functionality of the genome portal for this species is that we have an embedded JBrowse genome browser for each species. So essentially, this genome browser provides an interactive and user-friendly genomic visualization. Next. So some of you might wonder, okay, so what is new here? Because we have these very already robust global resources or genome browsers such as UCSC or Ensemble. Well, unlike these global resources, the aim of the Swedish Reference Genome Portal is to tackle and foster data sharing for local and specialized communities. And essentially what we do is we motivate people to make this data available in the global and the global repositories. But what we're doing is facilitating access for these local communities that might not have the bioinformatics competency to go to these standard repositories. So in this case, for example, we are targeting the biodiversity and evolution community in Sweden, but also we have people from the pathogens community that they would like to build their own genome portal. So essentially facilitate the access for that data type or that community. Also something that I'm very proud of is that we are encouraging researchers to make their genome annotations public, because actually very rarely people do that. And that's quite sad because, as we know, all the genomic annotations provide this additional layer of understanding of new sources of data. And unfortunately, there is not a single repository that aggregates this data. So what we do is we help the researchers. We suggest where they deposit the data. But essentially, we want to highlight genomic annotations that actually are derived from population or comparative genomics fields. So for example, anything that you can map and you have the genomic coordinates. So for example, FST metrics or nucleotide diversity, the GMSD, conservation scores, you name it. So we are helping researchers to make this data available, not only for the Swedish community, but also internationally. And all this while ensuring compliance with FAIR principles. So for us, this is an opportunity to start like training our community to start adopting the FAIR principles, essentially, to make sure that their data is going to be reused. Next. So how does this work? We have three main actors in this data flow. So we have the users, public databases, and the national infrastructure. So the users, we have the primary stakeholders, which are the researchers, universities, research institutions. And we have extended stakeholders, which are government, industry, and the general public. So how this process starts is like researchers that they own the data, they produce the data, they get in contact with us, the national infrastructure, in this case is Scilab Lab, and then we have already a built-in ecosystem, compute ecosystem that allows us to programmatically fetch information from public databases, so such as ENA, NCBI, Figshare. So that's for the genomic data. For species and genomic assembly metadata, we also fetch information from GBIF, Swedish database, GOAT, the UICN Red List, and the Swedish biodiversity data infrastructure. And then we build the website and put all the data in the genome browser and then make it available for everyone. Next. Yeah, you can go quick. Yeah. You can go on to the next one, so then it's all displayed. Good. So we built a system thinking on simplicity, openness, and easy maintenance. So it's simple in the sense that we use Hugo, which essentially is a static site generator. So that means that there is no need for a database or for a heavy server to build this web page. These web pages, or in general, the web platform, is lightweight and fast. So basically, you just have a collection of pre-built pages. Imagine like Markdown, and then you just build the information and you put the links. So it's as simple as that. And it's open and reusable. Essentially, all the code is open source, so it can be easily adapted to any application. And it's easy to update. So you want to add a new species or you want to update some data sets, you just update the config file. So it's very easy in that sense. And also we thought about portability. So in this case, we use Docker for deployment anywhere in any systems, and including Kubernetes cluster, that is the one that we're using. Next. So in summary, the Swedish Reference Genome Portal provides a centralizes the genomic data from local specialized communities, allowing users to easily discover and explore them in one place. It lowers access barriers, enabling specialists and non-specialists to better use genomic data, and it's simple, fast, open source, and scalable, and it provides a reusable model, so it can be used by any other institution, research group. Next. And with this, I want to thank you all for your time and attention, and here you see a collection of the QR code to our repo. If you want to know more about Scilab Lab Data Center, all the presentation slides are available in Figshare. Thank you very much.
Okay. Yes, okay. ¿Necesito usar esto?
Sí.
Ya, ¿y cómo es esto? No. No, okay. Okay, so good afternoon, everyone. I will try to keep this very short. My name is Juan José Guadalupe. I'm from Ecuador, the little country next to Colombia. So my presentation today is about the microbial communities associated with a cushion plant that is called Plantago rigida throughout microsuccession process in the Ecuadorian Andean Páramo. So first, I have to talk about the Ecuadorian Páramo. For those who don't know about the Páramo, it's a biome that it's between 2,800 meters to 4,800 meters, so it's really high. It covers, in the case of Ecuador, almost 6% of the whole country, so it's a lot. It's considered to be really cold, humid, and it's also intense solar radiation during the whole year. It has high endemisms of flora and fauna, the Andean condor, the Andean bear, that little plant that, I don't know the translation, but it's called chukirawa. It's important because it also provides ecosystem services to the highlands in Ecuador, to the main cities as the capital, Quito. It provides fresh water. The Páramo works as sponges for water. And it also captures CO2, so it's important in a climate change environment that we're living. So 20% of the Páramo are peatlands. Peatlands are waterlogged soils. There's a slow decomposition happening in these waterlogged soils. There's carbon accumulation happening. So under these extreme conditions, plants have to adapt. One of the adaptations, and this adaptation has also been seen in arid conditions or alpine conditions, is to form these cushion-like structures. So they have like these compact leaves and really deep root systems. So everything is covered by this cushion plant. In the case of Ecuador, Planta gorrígida dominates the Andean peatland. It grows from 3,000 to 5,000. It contributes to carbon cycling. It facilitates the growth of other plants that grow underneath it. But we have seen that Planta gorrígida undergoes to a ecological microsuccession. The causes and the consequences of these microsuccessions are poorly understood. We don't really understand. And the microbial diversity associated with this succession is unknown. So there's images of what happens with the plant. The first stage, you only see Planta gorrígida. The second stage, you see Planta gorrígida being colonized by other plant species. And by the third stage, you see that there is almost complete colonization. So the objective of this study was to examine how microbial diversity changes across Planta gorrígida microsuccession stages and how these shifts can influence the carbon cycling in the Ecuadorian Páramo peatlands. That's the Ecuadorian Páramo. So what we did, we took samples from the 3 stages that I previously mentioned. In the Parque Nacional Cayambe-Coca, we took samples at 4,000 meters above sea level. So we took a 60 centimeter sampler in the top of the Planta gorrígida, and then we took the first 10 centimeters to extract DNA. We extracted DNA, we sequenced, we assembled metagenomes, and then we did composition diversity and functional analysis. And also we took measurements of the gas emissions that were being emitted by the Planta gorrígida. Next. So as results, what we find is that, first of all, bacteria are the most abundant and diverse group associated with the plant. As you can see with the proportions compared with archaean plants, bacteria, it's almost 98%. By diversity, it's also the most diverse group. But as you can see, in every stage, it kind of remains stable, the composition. In the case of plants, it's more interesting. We found that there's a decrease of Planta gorrígida by stage 3, and we also saw that morphologically. We saw that in the case of grasses, Hordeum, Lolium, and Sia increases throughout the process, as you can see in the figure. This may correlate with a decline in carbon sink capacity, as we measured also with gases. The plant composition tends to change during this succession. In the case of archaeal communities, we saw that there's more changes by stage 3. We saw an increase of methanogenic archaea, like Methanobacterium and Methanocella. And as you can see in the emissions, by stage 3, there's also more methane emissions by stage 3. And this potentially is contributing to methane efflux and indicating shifts in microbial community is linked with carbon dynamics. And the conclusions of this part of the study, this is the first part we are building this baseline, is that this is the first study to describe the microorganisms that are associated with this plant. The the microsuccession process is changing the plant community and the archaeal community. And this could totally have a correlation with the methane efflux that we're seeing. And of course, there's more studies that are needed to explore these environmental factors driving this succession. And lastly, I want to thank my team, my laboratory, and if you have any questions, feel free to ask. I was missing that my team is working a lot in the mainland Ecuador and the Galapagos. So if you're interested in Ecuador, just feel free to talk with us.
Thank you. Thank you for the nice presentation. Thank you for the nice presentation.
That's what the
Oh, sorry. Thank you for the nice presentation. I don't know where the microphone is. Thank you. I wanted to ask you, how oxic or anoxic is the system? The whole time?
Noxic. So I'm also missing a lot of results. We also have like predicted functional genes, and we correlate them with the emissions and the reads that we got. So we're going to publish this soon. So.
I'm gonna try the clicker, but we'll start with it. Great. Thank you so much, everybody. As you said, my name is Jordana, and I work for African Parks that's based in South Africa. And today I'm going to be sharing with you a very exciting initiative that we have going on using eDNA across our protected areas to actually answer management questions. So some applied action here. Let's see, I'm going to test this. Oh, it did work. Okay, great. So African Parks is the largest conservation management organization in Africa. And we have pioneered the concept of a public-private relationship with governments and communities to take on the full responsibility of managing their parks. So a lot of times there's not enough funding within these settings, and there's a lot of paper parks or degraded parks. And African Parks creates this relationship where they take full responsibility and will help to restore these parks back to their natural state. We are currently in 24 different parks across 13 countries, managing over 20 million hectares with the great ambition to go to 30 million by 2030. So we will see. All right. So we've been measuring impact by numbers using species counts and abundances, number of snares that have been removed over these systems. However, we've gone through a paradigm shift where we're trying to move away from counting species to actually measuring ecosystem complexities and the functioning of these ecosystems. And we're going to be doing this through using species interactions. So species interactions are really the true architecture of biodiversity and the foundation of a functioning system. We say it all the time, everyone is connected, everything is connected, and that is so true, from below ground connections to above ground. And that is what we are going to be attempting to connect through food webs. Food web complexity, it really supports ecosystem resilience and functioning of these systems. So the more that we understand how everything is connected in these systems, the better we can manage them. And one way to do that is to use ecological network theory analyses to measure and quantify these interactions that are taking place in our environment. There was an elephant there, it disappeared. And that's because I just want to point out, it's not just the elephant that has left. Sorry, can we go back? Back to there, that's perfect. Okay, everybody saw the elephant leave. But it's all about the knock-on effect that it has throughout that entire ecosystem, right? It's not just that extinction, it's the co-extinctions that are taking place, and how do we capture those co-extinctions? One thing that's really important for African parks is they're doing a lot of rewilding. So they're actually doing the opposite of this. They're putting species back into systems that haven't been there for maybe 20 years. So how is that system going to rewire and interact with its new ecosystem? So we're hoping to also capture that. Okay, now we can go to the next one. Thank you. And we haven't been able to really do that until today. Not today, but the past couple of years, with the onset of DNA metabarcoding. Everybody is well equipped with understanding that in this room. But what is most powerful for us is the accessibility. Anybody in the world can collect a sample and contribute to conservation. And that is very powerful in these remote areas that we're working. So from scat samples, we can help to reconstruct these food webs and soil and water samples. Next, please. So combining these two things, the molecular side and the ecological network analysis, we have this kind of holistic approach to attempt to measure ecosystem integrity because we can capture the structure, function, and composition of these systems. So we are setting out to do we are currently running a pilot study across 5 parks. I don't know if you can see the stars, but we're trying to do it across diverse biomes from the deserts in Iona down in Angola to Edzala in the Congo, and the tropical forests to the savannas, from new parks that have been acquired by African parks to old parks that have been in the system. So we're going to be looking at 5.5 million hectares that we are assessing in this landscape. And just want to point out that's of the parks, but we're also looking outside of the parks in the unmanaged areas as a comparison reference point to what we're measuring. And this project is a 2-year period, very short, and we have about 5 months left. Thank you. And then please just go ahead and click through these. So our objectives are to measure baseline biodiversity using fecal, soil, and water samples, characterizing diet diversity and composition through carnivore, herbivore, and omnivore scat samples to reconstruct the food webs, and then characterize soil diversity using shotgun sequencing, and then pulling these all together to create this park level evaluation to look at ecological integrity and impactness across these systems. And most important is to enhance capacity of African science within these regions. We are working with PhD students, master students, local universities, and creating these workshops within each of the countries to train new students on these techniques. Thank you. So we had a very successful year last year. We only started in March, and we ended up with 8,000 samples across these 5 parks, averaging about 80 field days for each of the parks across 4 months. And we targeted the dry season for each of these parks. Next, please. This is a lot of numbers. All I want you to take home from the sample collection is that we ended up with over 100 different species that we collected fecal samples from for diet analysis. The soil samples we collected across different habitat types and burning regimes, because that is what's important to management, is understanding the impact of fire on their soil, and then water samples across the rivers and lakes throughout each of these park systems. Again, a lot of numbers, but just to showcase, we did end up getting all the samples out of country with all the permits and working with the governments on this, which is a big feat. I'm sure a lot of you can appreciate that. And our sequencing of all the fecal samples have been done. We used the vertebrate 12S primer to capture the predator-prey interactions and the TRNL for plants. And we ended up with over 240 different prey taxa and identifying over 25 new species to the park systems that were not currently on their park species list, which is exciting. And that ended up with over 1,000 predator-prey species interactions and 50,000 herbivore plant interactions across this landscape, looking at about 800 different plant species. So just wanted to touch on a little bit of the data. He says, no, I can't. Can I talk about it a little bit more? Okay, great. This was a sanity check. So this is an NMDS plot. What you need to know here is each dot represents a dung sample. The closer they are together, the more similar their diet composition is. So what we can see here is looking at forest elephants and savanna elephants, they are very different, which is great. That's what we want to see, divided by that vertical axis. And that even the savanna elephants, they all split out according to their parks, which is what we had hoped to see because of the diversity of vegetation in those landscapes. A quick fingerprint of looking at our predator species. We get a quick snap of what lions are eating across all of our parks and who are the key species in lion and leopard here. Just blow through this. And now looking at patterns within parks, which is our main focus here, is we can see how the axes split out between the browsers and the grazers. And most important for us is looking at niche overlap, because it's been found and correlated that systems that are more intact have a greater niche partitioning taking place in that. So it is an indicator of ecosystem intactness. And then this is just looking at diversity within the parks, just from the diet analysis. We can look across, their common breadth to their total diet richness of the species. This is in Kafue National Park. From the top, we have our grazers, very specialized and narrowed, down to our broad consumers of elephant and impala on that side, and our cheetah, very narrow at the top, down to our leopard, which are more generalists. So really showcases our generalists and our specialists within the group. You can hit the next one if you want. I just find this interesting. We're also looking at all the wild, not only the wildlife, domestic animals inside the national parks, which is also always a topic of concern, right? And in Iona especially, we have a 5-to-1 ratio of domestic animals, actually 5-to-1 wildlife. But what we see, you hit that next one, please, to be aligned, they really diverge. When they're in the same habitat where there's absolutely nothing to eat out there, this is really wild, they're actually splitting apart in their diets, which is quite interesting. But we have a lot of cattle samples if anybody's interested in domestic livestock across Africa. Thanks. And then that brings us to the ecological network, which is a work in progress. We're busy constructing these at the moment with Dr. Pyers. He's based out of Unicamp in Brazil, who's working on all of our network analyses. There's many metrics that we can get out of this and ask for management to identify our keystone species, our vulnerable species, understand the robustness of our systems, and what's most important is, measuring all of these direct and most important indirect interactions that are taking place in that landscape. So we can look at how wild dock are impacting the acacia trees within Kafue. Thanks. And then this is just touching a little bit on the fecal samples, but we have all of the soil data and the water data that is actually coming out this month. So we are going to have a ton of sequencing data very soon that we will compile and compare across parks. And the idea is to do a feasibility analysis to see, is this the way forward for measuring impact, and to compare this over time. So this will be creating our baseline, and then every 3 to 5 years, we can come back and redo this assessment across our parks. Next slide. And then the big reason for us to be here is be expanding our network and looking for collaborations amongst this community and getting guidance on best practices. So we've been learning a lot about how to input our data now into GBIF that we're working with. So if anybody would like to help or collaborate or could use any of this data, have students that would like to use some of this data, please do find me. I did the next one. So thank you all very much for listening. Looking forward to hopefully hearing from some of you. And a huge thanks to all of the rangers and researchers and NGOs and community members that helped to make this all possible with collecting these samples across these vast spaces. Thank you.